Welcome to iVOM. This is the fourth webinar of our webinar series entitled Take a Walk at the VOM site. This is a joint organization of the Virus of Microbes 2020 Organizing Committee, the ISVM, and the Phage Directory. We have started this webinar series in September this year, and we will continue until February. Today, the topic is virus-host interaction molecular mechanisms with three outstanding speakers. And before passing the word to our lovely session chairs, Karen Maxwell and Julia Frunzke, let me just remind you that this webinar series is not replacing the real Virus of Microbes 2020, that we are still planning to be a face-to-face meeting in July 2021. I also would like to remind you to use the chat to ask questions, and please unmute your microphones and switch off your cameras during the session. On behalf of the organizing committee, I'd like to thank all speakers, chairs, sponsors, and the audience for helping us making this a successful event. I wish you a good session and a Merry Christmas and a Happy New Year. Karen, the session now is yours. Thank you.
Thanks very much. My name is Karen Maxwell. I'm a faculty member at the University of Toronto in Canada, and I'm delighted to be co-chairing this afternoon's session of Viruses of Microbes that's focused on the molecular mechanisms of virus-host interactions. So we'll be hearing from three of my favorite phage biologists this afternoon, Rotem Sorek, Peter Fineran, and Rob Lavigne. And speaker in this session is Peter Fineran, and he's from the University of Otago in New Zealand. And Peter, of course, has done a lot of work studying the adaptive immune system, the class 1 CRISPR-Cas systems, and he's going to talk to us today about some of that work.
Perfect, thanks Karen, and thanks to the organizers for putting on this conference. It's great to actually get everyone together, even if it's virtually. So today I was going to talk about some work we've been doing to understand how CRISPR-Cas systems are regulated. And so most of you will be quite aware of what CRISPR-Cas systems are and what they do. So these are adaptive immune mechanisms within bacteria and archaea. And they are composed of these Cas genes, which provide the enzymatic machinery, and the CRISPR arrays, which provide the immune memory of these systems. And what happens initially is an invader element will introduce its nucleic acids, and these are sampled and added as new memories to the CRISPR array. These are then transcribed and processed into guide CRISPR RNAs, and then these form complexes with these Cas proteins, and then these carry out a surveillance function and identify invading elements, and then degrade these and protect the cell. So a lot's been done in recent years to try to understand the mechanisms and also exploit CRISPR-Cas systems, but a lot less is known about how bacteria control their adaptive immune systems and when they might turn on and off immunity. So why would we actually be interested in that? Well, actually, We know a bit about that due to some really nice work from Rotem Sorek's group about 10 years ago. So why are CRISPR-Cas systems regulated? They actually showed that within bacterial genomes, there's evidence of spaces that match the host genome itself. In fact, our group and other groups then subsequently showed that experimentally we could identify CRISPR-Cas systems acquiring spaces from their own genomes, and this then led to a toxic, self-destructed autoimmunity. There's also some evidence from Luciano Marafini's group and colleagues showing that CRISPR-Cas systems can also be detrimental, for example, when they inhibit the acquisition of beneficial plasmids, and that's something I'll come back to. And there's also evidence that there can be just general costs for carriage of CRISPR-Cas immune systems. So to study regulation, we've been using this sort of our favorite model organism recently, Serratia, which was found in a salt marsh in New Jersey. But why we love it is because it has 3 different CRISPR-Cas systems in the single strain. It has 2 type 1 systems, a type 1E and a type 1F, both of which target DNA. And it also has a type 3A system, which targets both RNA and DNA to provide protection. So this enables us to look at regulation of multiple systems in a single host. But what we realized is while we were trying to understand about regulation in bacteria, we realized there was a complete lack of tools to study at a genome-wide level regulators and to identify regulators that control a particular gene of interest. So that's where we managed to recruit a really talented PhD student, Leah Smith, and she worked really hard to develop a non-biased genome-wide approach, which we're calling sort and seek, to identify regulators. What Leah's actually done is she takes the promoter of interest, in this case from the type 3 system, fuses this to a fluorescent reporter, and then the idea being that the reporter expression will indicate the levels of CRISPR-Cas activity and will be either activated by activators or repressed by repressors. And we then carry out a transposon mutagenesis to generate hundreds of thousands of mutants. And with each individual cell, there'll be a specific mutation and a different level of fluorescence depending on whether a regulator is mutated. And then we use fluorescent activated cell sorting to basically sort cells into different fluorescent level bins. So we can isolate those that have low level of expression, those that have high levels of expression, and we also sequence the other categories. We take the DNA from these samples, we then sequence across these transposon junctions to identify what genes are mutated, and by mapping these back to the genome and looking to see how many of them are present in particular genes, we can identify genes that might be enriched in low fluorescence pools, for example, to represent activators. This has worked really well. So in this particular study, what Leah's managed to do is generate about 300,000 mutants throughout the genome. And these seem to find about 30 genes which are present, or genetic loci that are enriched in either the low expression pool or the high expression pool. So we want to actually know whether these mutations represent genes that are actually affecting CRISPR-Cas regulation. So by taking single mutants and then looking at their effects on CRISPR-Cas expression, what Leah could show was, in fact, this method is able to identify regulators of interest. So we're really excited because we think this is now a tool you could use for essentially finding regulators of any gene of interest in pretty much any organism which has fluorescent reporters and transposons. But I'm just going to focus on one, which was the one that had the lowest level of expression in these studies. And this is a gene called IgAA. And it turns out that IgAA is actually part of a RCS membrane stress response pathway, which is well studied in E. coli. So this pathway actually responds to outer membrane, periplasmic, and peptidoglycan stresses. And these can be induced by things such as antibiotics. And what happens is that under normal non-stress conditions, this IgAA protein will actually bind and inhibit this downstream phospholipase pathway and block its activity. However, upon sensing stresses, this lipoprotein RCSF inhibits IgAA, and that allows this phospholipase to be activated, and these transcriptional proteins will actually turn on or off to target gene expression. So to actually verify that this is what's happening, Leah mutated the IgAA gene and showed that the type 3 system had reduced expression, and that by introducing subsequent mutations in this downstream pathway, this alleviated that effect, showing that pathway was involved. What she went on to do was to then show that, in fact, CRISPR-Cas interference by this type 3 system is also affected. And we do this by looking at conjugation assays. So we take a plasmid which is targeted by the CRISPR-Cas system and we conjugate that into Serratia. So if we have untargeted controls, the bacteria take up the plasmids very effectively. If the wild type system targets the plasmid, we see a strong reduction in plasmid uptake. And surprisingly, the IgA mutant had almost completely abolished type 3 activity. What we then saw is that in fact, type 1 systems were also affected in a similar way. So remember, this has 3 different CRISPR-Cas systems, and we saw that type 1E expression, type 1F expression, and also interference were all impeded by this RCS pathway. So it's showing that the type 1s and 3 systems are all regulated. We then also observed that the adaptation of the CRISPR-Cas systems was affected by this RCS pathway. So we can see here, this is the acquisition of new spaces by the CRISPR-Cas immune systems. In the IgA mutants, you can see a reduction in space acquisition in both the type 1E and type 1F systems. What we can see as well is that actually the acquisition of spaces is necessary to drive out plasmids from the cells. And when this pathway is mutated, we see that the cells are actually unable to drive the plasmids out more easily. So this is leading to essentially a reduction in adaptation and a reduction in ability to cure plasmids. So what I've shown you so far is we discovered that RCS is involved in inhibiting the CRISPR-Cas pathway, which is allowing plasmid uptake and retention. But then we're really curious to know, well, what's actually happening with phage infection? So what Leah first did is she took 4 different phages that are unrelated in our collection and tried to infect the wild type. And as we expected, they all infect very nicely, shown as these curves. And when she took the IgA mutant, in the absence of any CRISPR-Cas immunity, we found that it was actually resistant to all of these unrelated phages. So there was a pan resistance. So there appears to be sort of a CRISPR-independent immunity that this RCS pathway is triggering. So to get an idea of what was going on, we took a look at the cells. And in the wild type, we see that they're flagellated. And this is an important receptor for a number of these phages. Whereas this IgA mutant actually has lost flagella and has some altered cell surface properties. And this is very commonly known for a lot of RCS mutants in Enterobacteriaceae. And it turns out that 2 of these phages that use different receptors actually have significantly reduced their ability to bind to the cell surface. And this has reduced their absorption. And this is because, in fact, the receptors for these 2 phages are downregulated by this RCS pathway. So what we're seeing is a CRISPR-independent phage resistance that's being stimulated by this RCS pathway. So what we actually think is going on now, if we put this all together into sort of our model, is that we think that this RCS stress response pathway is inversely controlling both surface immunity and CRISPR-Cas adaptive immunity. And by doing this, it's enabling the bacteria to sort of discriminate between plasmids and phages. So under a non-stress condition, where we have CRISPR-Cas system activity, we see that there's a degree of phage resistance as well as resistance to horizontally acquired elements such as plasmids. And whereas under stress conditions, such as what can be triggered by antibiotics, we see that there is a loss of phage receptors and altered cell surface properties that leads to a pan sort of innate surface resistance. But at the same time, this RCS pathway is repressing CRISPR-Cas, which is enabling cells to both acquire and retain plasmids more easily. So we think this might allow cells to sort of be more flexible in sampling some of that genetic material that's out there and potentially acquire beneficial genes. So I'd just like to finish really thanking Leah, who drove this project and has been a fantastic PhD student and now staying on as a postdoc, and all of the members of the team in the lab, as well as generous funding from all our funders. And looking forward to having some questions at the end. Thank you.
For this little gap before. My name is Julia Frunzke, and our lab is at the research center in Jülich in Germany, and we are interested in bacterial networks and phage-host interaction in actinobacteria. And it's my pleasure to introduce the next speaker to you, who actually probably doesn't need an introduction, Rob Lavigne from the KU Leuven. And yeah, we'll just take 5 to 10 minutes maybe. And I will talk, Rob will talk about the molecular hijacking of bacterial cells, and he's interested in the diversity of phage-encoded mechanisms that modify replication, transcription, and also in post-translational modifications within the cells. So Rob, we are looking forward to your talk.
Thank you. Do you see my screen? And do you hear me?
Yes.
Okay, thank you so much. Thank you a lot also hosting us, and of course, Karen and Julia, always nice to see all of you. I'm going to talk very specifically what interests me the most. I think phages are nature's first bioengineers. When you look at the lytic infection cycle, it's for me just amazing how in a few minutes the host takeover mechanism happens, and this fully adapted mechanism by which specific phage proteins and bacterial proteins interact with each other to convert the bacterial machinery into a virus producing machine. I'm hoping on one hand by mimicking these proteins to small molecules that have an antibacterial effect, an antibiofilm effect, an antivirulence effect. This could lead to a new generation of antibiotics. But the phage proteins themselves might have some applications in synthetic biology and so forth. So our lab has been dealing with this, I realized, for over 15 years now, and together with other amazing groups like Joel Bondi-Denomi's group, focusing on mechanisms that disrupt the bacterial mechanism in Pseudomonas aeruginosa, where you have at transcription level, at metabolism level, cell division and replication mechanisms that are being hijacked and blocked by specific phage proteins. And today I'm going to talk about one story, in the interest of time, of our protein IgE from bacteriophage LUS24. This LUS24 is a temperate phage, so it's not suitable for phage therapy, still containing a lot of unknown ORFs, and these are the ones we usually tend to look for, the ones that are most likely to modify the bacterial metabolism in specific ways. And our general strategy is to lift this phage ORF out of its natural environment of the rest of the phage genome and simply clone it into an expression vector, expressing it as a single copy from the chromosome of Pseudomonas aeruginosa, and see what kind of changes phenotypically this elicits within the bacteria. This is, of course, a step away from the natural biology, but it's teaching us a lot in terms of how its toxicity is working. And for example, for these early proteins for LUS24, we found one specific open reading frame, IgE, which when the gene was not expressed is simply growing, but when you induce the gene by IPTG, there is no bacterial growth. And for this specific protein, it was interesting to see that Pseudomonas was being inhibited, but actually E. coli was not inhibited. So this phage protein, IgE, had a specific effect against Pseudomonas aeruginosa. And when we look under the microscope, you have a normal forming microcolony. Induction of IgE resulted in this elongated cell growth, which is indicative that either replication is being hit or cell division is being perturbed in some way. So we followed this up by understanding at the molecular level how this is happening. I think, yeah. And in the first step, we saw this protein is 48 amino acids long. Yeah. So we made various deletions and actually saw, yeah, we could delete only the last amino acids to have still partial toxicity. So we're talking about IgE is a small peptide of 48 amino acids that are all necessary to retain that toxicity. So not much flexibility in making it shorter. Of course, we wanted to know what is the target of IgE, what bacterial protein or other component is it interacting with, and for this we did a straightforward pull-down assay in which the lysate of Pseudomonas aeruginosa has passed over a column with glutathione, and which is loaded with GST fused IgE. And then we check which protein is being retained, which bacterial protein is being retained. And the protein we found, one candidate protein, was the gyrase B protein, which is part of the DNA gyrase complex. This is, of course, let's say, an in vitro assessment, an in vitro interaction that you're capturing on a column. So we usually try to confirm this in an in vivo way, and bacterial to hybrid is our go-to mechanism. And the basic principle is that you have this adenylated cyclase producing cyclic AMP, which is produced in separate domains. And only if there is an interaction between IgE and the gyrase B, there is the production of cyclic AMP, production of which induces the production of a reporter gene, simple blue-white screening, basically. Yeah. And doing this by cloning various fragments of DNA gyrase, we were able to confirm actually the interaction between IgE and gyrase B, and really lock it down to its interaction domain within that protein. Which actually confirmed nicely our observation under the microscope that DNA replication was being hit. So we wanted to actually see what's happening, what is the function of gyrase. Well, simply put, it's introducing negative supercoils. So in a simple in vitro assay, if you take some DNA and you add gyrase to it, it will become supercoiled and will migrate more quickly across a gel. An inhibitor of the gyrase, for example, the important antibiotic ciprofloxacin, if you add an increasing amount of ciprofloxacin, you see that there is less and less supercoiling, and the same effect we saw when we added our phage protein. So that's where the name is coming from, IgE, an inhibitor of the DNA gyrase, and it's a small peptide confirmed by these functional assays. From a biological perspective, and when we look at blocking, obviously in the presence or absence of ciprofloxacin, we saw that although there is an obvious decrease in the plaque-forming units when there is ciprofloxacin present for this phage, it still doesn't strictly require the bacterial DNA gyrase. So even when the DNA gyrase is inhibited, still the phage has some efficiency left, while other phages are completely reduced in their plaque-forming capacity, which I think is also going to be relevant in the choice of phages that you use in phage therapy, especially if it's combined with antibiotic treatment. But in any case, it shows quite logically that an early phage protein is blocking the bacterial replication because it doesn't need it to complete its replication. So when we look at the DNA gyrase complex, it's important to realize that this is indeed a very important antibacterial target. As I mentioned, it introduces these negative supercoils, and it does so as a protein complex between gyrase B and gyrase A. And ciprofloxacin and other fluoroquinolones are acting against the gyrase A part of this protein complex. and our phage protein is actually interacting with gyrase B. So we were interested to see if there was a separate or independent working mechanism between the two. And one way to look at this is by actually taking bacterial mutants that are resistant to fluoroquinolones. These are well-established specific mutations that introduce resistance to fluoroquinolones. And you see that the bacteria grow happily in the presence of this ciprofloxacin. And we were wondering, if we introduce our phage ORF here, is it still going to be toxic? And indeed, that is the case. So that suggests to us that there is no cross-resistance mechanism, let's say, between the ciprofloxacin and the Ig peptide, and therefore it's an indication for a different binding mechanism to this very powerful and important antibacterial target. So that's obviously only part of the story. We recently published a review article on the different strategies you can use of applying these small phage ORFs, eventually leading to small open reading frames. And of course, there are plenty. And hopefully, because these phage proteins are fully adapted to the host and acting independently, orthogonally from it, it can inspire antibacterial design strategies as well as biotechnological applications. So if you have a phage, even if it's temperate, perhaps it can be the basis for small molecules inspired by phage that have these antibacterial or antivirulence effects. Obviously, at the same time, on the negative side is that some of these stories take a long time to fleece out, and you have to start connecting to some powerful chemists who can start mimicking this phage protein into these small molecules that mimic the effect of the phage protein, and therefore serve as inspiration. But hopefully it can be yet another layer of antimicrobials next to lysins and biofilm depolymerases as enzyme antibacterials, and phage therapy, natural phage therapy, as well as bionanoparticles engineered and equipped with antimicrobials. So this work is currently under review. I want to specifically acknowledge Kostya Severinov for this collaboration, and Professor Aartsen within KU Leuven, who does all of the microscopy. Apart from the paper, we're also having a music CD brought out on this phage-based inhibitor, but that's probably only for the 40+ people among us. And there, for the rest, I want to thank my entire crew, united apart, I would say, and just like us, again, I want to reiterate that we may not be together physically, but in spirit, it's great to be with you guys. Thanks.
All right, that's great. Thanks, Rob. That was a fantastic talk. Well, that's great. So I am going to hand over to Julia now to address the questions for the next two speakers.
Okay, thank you. We have a few questions now for Peter Fineran. One from Giovanni Denomi. Peter, given the screen was probably done in the absence of phage, do you think that whatever antibiotic you were using actually triggers the stress sensing and Crispr regulation?
Yes, that's quite an interesting idea. I mean, what we verified the mutants afterwards as well in the absence of antibiotics. So we're certain that it wasn't purely an antibiotic stress. And we also identified multiple mutations, for example, in OPGG, OPGH. These are proteins involved in osmoregulated periplasmic glucans, and these are important for like osmotic stress and things. So when those are mutated, you get stress in the periplasm, and that's triggering this stress response pathway. And in fact, the IgA mutant is actually, that's triggering the stress response. So we actually the experiment is actually happening in a non-stress state, and we've identified mutations which are triggering that stress response. But it would be lovely to do it in the presence of phage infection as well. There's probably a few challenges we'd need to iron out technically around getting the phage infection to happen and the cell sorting and cell survival and that sort of thing, but it'd be great to do.
Sure. Okay, and the next question would be, the gene WZT appeared to be highly expressed. Is it involved in LPS transport? Somebody's wondering whether there's a link between CRISPR-Cas system and the LPS transport in general.
Yeah, so the WZT gene, this was highly enriched in terms of insertions in that pool, and it's quite well known to be involved in LPS transport. So again, these mutations are probably causing a stress at that, the outer membrane. And there's also evidence that LPS mutations can trigger the CPX stress response. And one of the other genes that we identified in the screen was in fact CPXA as part of the CPX pathway. So we think there's also, additional stress response pathways that may be sensing, things that are happening at that membrane. So alterations to LPS, alterations to the periplasm, etc.
Yeah. Okay, and then there is a question whether you think there could be a link, or CRISPR-Cas systems could be linked as kind of two-component systems.
Yeah, I think that's a nice idea. I mean, there's a few more studies that are sort of starting to emerge of CRISPR-Cas systems that they don't only operate sort of in isolation. So you can have, for example, sharing of adaptation machinery between CRISPR-Cas systems. You can have sharing of CRISPR RNAs between different CRISPR-Cas systems. So there is some sort of, I guess, linkage between CRISPR-Cas systems. Whether there's sort of signaling hierarchies between CRISPR-Cas systems, that would be very interesting to look into. I'm not sure that there's any direct evidence of that yet.
Yeah, neither. Okay, and the final question would probably be if you think there is actually a link between RCS and competence, so suggesting that actually under stressful condition, cells can scavenge important DNA.
Yeah, I know certainly in other bacteria, the RCS pathway, it does regulate some of the competence genes. I wish that was the case in Serratia, because this strain is, very recalcitrant to transformation. So we have to do everything by conjugation essentially. So we don't have competence in this strain, but I think in other strains where the RCS pathway is present, this could be involved in competence. In fact, when we did some data mining as well, we found evidence of the RCS pathway also regulating CRISPR in a Winnia strain as well. So we think that this result actually goes beyond just this one example. It's probably happening in a number of other bacteria as well.
Mm-hmm. Okay, thank you very much. I think that have been the question from the chat I collected for you. Thank you very much for being here. So we move on to Rob. Actually, he already did quite a busy job in the chat with answering questions, but I will just repeat them, right? Sure. So I will start with my own questions. So do you think that the Ig peptide is now a good basis for developing an antimicrobial peptide which could also be used for treatment? I mean, is it too large?
It is large. 48 amino acids may still be too large, but we don't fully know. As I responded in the chat, the gold standard would be to have a cryo-EM reconstruction of the entire complex where the phage protein is bound to it. And that would allow assessing whether it's possible by chemists to make small molecules or peptidomimetics that go. But I think it's worth the search in this case, because the DNA gyrase is such an important antibacterial target.
Yeah. It's nice actually, because there's just recently been a cryo-EM structure of the gyrase complex, right? So this could be interesting.
Okay.
Yeah.
We will see if we can do some modeling. For which bacteria?
I think it was E. coli, like always.
Yeah.
Okay. And just one more question. So if you overexpress the Ig peptide, how long do the cells actually suffer from this inactivation? I mean, how long do they take in liquid culture to, form suppressive Did you test this or?
Any mutations that would and usually you have to in the bacteria, so it takes quite a lot of triage to find, on one hand, the phage mutations that stop the activity, in this case of IgE, which is useful information, but of course you also want to establish bacterial mutations that suppress the toxicity, but in this specific case we haven't found them.
Okay, thank you. So we move on with a few questions from the audience. What about the homology of IgE with other Pseudomonas phages and phages of other species? I mean, is it kind of a conserved peptide you find in many things?
No, so it's not conserved at all. So we do find it in the other related phages of that clade, all of which are, or most of which are infecting Pseudomonas aeruginosa. But it's one of these typical small, sometimes even overlooked, because it's smaller than 50 amino acids, which is the threshold some tools or people take in gene prediction. So it's not widely conserved. It's highly specific, and that also explains probably why it's specific to Pseudomonas and not inhibiting probably the E. coli gyrase.
Mm-hmm. And another interesting question: how does IgE contribute to host takeover? I mean, what's the function for the phage?
Yeah, our logic there is that it's expressed when the DNA replication of the bacteria should be shut down. So potentially that viral the virus has the benefit of harvesting nucleotides that are more available and not going in competition. That could be one option, but yeah, the disadvantage is that by taking one ORF, you lift it out of its natural environment. I'm hoping that Joe Bondi-Denomi's recent Nature Methods paper will enable us also to streamline the engineering of lytic phages to knock out or knock down or tag genes in a more efficient way, because that's really what's been limiting us.
Mm-hmm. Okay, and here we have a question. Do you think that the IgE peptide is working on itself? I mean, that's basically already commented on that. If you're taking it out of the natural network of the phage. In other words, do you recommend to investigate different combinations of phage early proteins, whether you have synergistic effects there?
Yeah, we sometimes do that. Expressing this phage ORF and then having a genome library of the phage also introduced in the bacteria to see if there is anything that complements that toxicity or suppresses that toxicity in the phage. But again, I don't think we did it for this phage protein.
Okay. Maybe final question to Rob. So, I mean, you tested this for the Pseudomonas gyrase, right? Or was it an E. coli assay, the supercoiling assay?
The supercoiling assay was just in vitro, taking DNA and the purified gyrase.
But from Pseudomonas?
Yeah.
But do you know how broad the activity profile would be? I mean, testing it to more, I mean, other related or far away gyrase?
Yeah, it's also in Putida, it's toxic, but not E. coli.
Okay. Thank you very much. I think that was most of the questions for Rob. Yeah. And with this, we are at the end of the session, and it remains me to thank all speakers again for their excellent contributions. And of course, to the organizers setting this up. And I'm very much looking forward to meet some of you back again in January, and maybe at some time in a real meeting next year. Until then, I wish you a Merry Christmas.
Okay.
Thank you.
Hi, everyone. Hi, everyone. It looks that we had a great iVOM again. So on behalf of the organizing committee, We would like to thank all the speakers for the excellent presentations, very nice indeed. And of course, to the chairs, Karen and Julia, for driving the discussion. And I will also would like also to thank the audience. We reach almost 300 participants, which is very good. And to remind the audience that you can watch again our iVOM sessions at our YouTube channel. You can see in this slide here. Let me call also your attention to the International Society for Virus of Microbes. If you are working in the field and if you are interested, you can join the society and become a member. You just have to go to the website and see the details to join the society. Let me just introduce our next iVOM, which will take place next month, again, under the theme Agro-Food, Veterinary and Environmental Biotechnology Applications. And we already have our confirmed speakers, which will be Danish Malik, Sam Newgan, and Ana Oliveira will be our speakers for the next iVOM. And before closing, and because it's Christmas time, we also want to wish you a Merry Christmas and an excellent New Year. We're hoping that the pandemic is overcome and then that we can see all of you in Portugal for our conference, our Virus of Microbes conference that will take place in Guimarães next summer. So goodbye to all. Thank you again for all the participation of the speakers, chairs, and the audience. Bye-bye.